Today I'm happy to share with you how we study development gliobiology to associate disorder. Particularly today, I'm going to share with you two different cell types in glia. In the green cells, oligodendrocyte. In the right, there's an astrocyte. So my lab is actually developed biology lab. So we use a chick in vivo system to identify novel glial genes and then identifying their conjugate role using genetic mouse model as well as injury model, which mimics specific disease angle. Because glia cells very abundant in everywhere, we study lots of disease, including white matter injury, stroke, and glioblastoma. So particularly today, I hope to share two diseases, but if time is limited, I will definitely focus on one of my core research paradigm in white matter injuries. So why we care about white matter? Because I am very glia-biased scientist. Glia cells are bodies located in this white matter tract, and then of course the neurons in the gray matter. If you look at in the cellular level, you see that neuron in this purple, which is a very important functional unit. However, you see lots of glial cells surrounded by the neuron. If you look at this in the electron microscope level, you see that oligodendrocyte, the cell produces myelin, surrounded by axon, have very nice multi-pushes of the myelin, called the myelination. Therefore, oligodendrocyte is very important for providing the metabolic support and increase electrical stimulation. And if this myelination is really impaired, it causes a lot of disease, which I will touch a little bit later. Another cell, this cell is called astrocyte, the most abundant CNS populations. And astrocytes really interact with a synapse, so it's important for the synaptic transmission. And also, which I didn't show here, but astrocyte endophil structure interact with endothelial cell to form the BBB, therefore it's very important for the metabolic support. When there's injury environment or disease condition, this astrocyte become very reactive, morphologically bigger, and they have a lot of dual functions. So my lab is particularly working on those two types of glial cell in both development as well as disease. So I was very ambitious to prepare two parts, but with the time limit, mainly focus on one part, but I hope to share with you some of the second part of the research as well. First part, we will talk about how oligodendrocyte development, what happened after injury, what signaling pathway is important. In the second part, I will also share with you what gene is important for the astrocyte development and associated disorder. So let's just start with the first part, myelination. As I mentioned, your oligodendrocyte is the cell that produces myelin in the central nervous system. And it is very interesting that myelination starts when we are born and continues to develop until like a young adult, age under 50. I believe in our group, everybody's still developing myelin in our central nervous system. And as you can see here, it is also noted that individual who has a more performance has a more decomyelination than normal individual like me. So this unique development paradigm really give an idea if there's insult happen. Oh, before that, this myelination is really important for the high level of CNS function. It is known that change of neuronal activity, motor learning, or social interaction change myelin thickness. And also very recently, if you manipulate myelination, they can also affect this high level of CNS function. This is evidence that not only neuronal activity important, myelination is also tightly associated with high level of CNS function. So given this unique critical function and unique paradigm of the development, if there's injury happen, disruption of this myelination can cause many different diverse neurological or neurodegenerative disease affecting in people in all age, from the baby who born prematurely or full-term baby under hypoxic condition, or genetic disorders, associated with the myelination defect and also autoimmune disease in the multiple sclerosis. But recently, late onset disease such as Alzheimer's disease or vascular dementia also cause lots of white matter damages. So this is kind of the area that my lab is really interested. And the reason why we have still problem with the myelination, that's kind of what my lab is trying to understand in the cellular and molecular level. As a developmental biologist, we always try to think from the early development, and oligodendrocyte lineage development is pretty well established. It starts from neural stem cell, and they specify proteome cell. As you can see, morphologically very well branched over the time. And when they are fully mature, they actually wrap around the axon, and then you see those nice, myelinated axon structure. And it's interesting in this one cell during this maturation process, beginning, they need a highly polymerization of actin. As you can imagine, they need to branch out. However, when they start wrap around the axon, they actually need to break down actin. So it is very interesting that one cell throughout their life cycle need to have heavily actin polymerization as well as disassembly. So this is kind of evidence that cytoskeletal remodeling is one of the critical parts for the oligodendrocyte development. So what else is known for the oligodendrocyte development as aspect of the signaling? Wind signal is quite well established, but as you can see, different arrows quite complicated. Wind signaling in very beginning stage need to be inhibited to specify from neural stem cell to OPC. Then when this OPC start proliferating, Wnt need to activate it. And then when they start to differentiate it, now they need to inhibit. So it's a very dynamic signaling. So that make a little bit difficult to study, but also very interesting to study. And this is like how we know about Wnt signaling in oligodendrocyte development, but it's also known that Wnt signaling in disease aspect. So this is MRI image from the multiple sclerosis, and the lesion is identified this white focal lesion. We look at this lesion a little bit carefully. There is also recruited oligodendrocyte, excuse me, progenitor cells, meaning progenitor cell can be recruited into the lesion area. They can possibly repair. However, this cell is kind of stored there, not properly fully mature, because people has found that this OPC, stored OPC, has high Wnt activity, and this high Wnt activity really inhibit oligodendrocyte progenitor cell differentiation. So long story short, Wnt signaling is important for throughout the development, but injury paradigm so far what people believe Wnt is kind of antagonize oligodendrocyte differentiation for the repair process. So with this background, what I'm trying to tell you that oligodendrocyte lineage development and repair process, cytoskeletal remodeling is important, Wnt signaling is important. So with that reason, my lab is interested in called DAM2. It's a very unique name, DAM gene. And this gene is a formin family member, and formin is important skeleton remodeling. And as you can see, the structure here, this is a GTP-based binding domain, has a two formin homologous domain. And then family has two members, DAM1 and DAM2, and DAM1 has been extensively expressed in the neuron. However, DAM2 expression is very restricted in the glia cell. That's one of the reasons that we are very interested in DAM2 function. Excuse me. So this very non-overlapping expression patterns such as different function. However, DAM1 function over 30 years has been very extensively studied in neuron, obviously in neuroscience field. However, DAM2 function, when I first touched on this gene, there's no characterization of their function. So my first investigation when I was postdoc, I want to know what is DAM2 function in glia, because they are very restricted expressed in the glia cell, both oligodendrocyte and astrocyte. So first starting point is we always want to know if we want to study novel gene, when and where this gene is expressed. So TEM2 is expressed in both spinal cord and the brain in oligo2 lineage cell. Oligo2 is a marker for oligodendrocyte lineage throughout all the OPC to mature stage. So we know TEM2 is expressed oligodendrocyte lineage. And with early stage work in my postdoc work using the chick spinal cord, which I didn't bring you here. Long story short, what does TEM2 do? They actually assemble this Wnt receptor complex and amplify Wnt signaling. So this is all what we know TEM2 function in early chick spinal cord development. And basically TEM2 is a positive canonical Wnt regulator. And as I mentioned you, if you remember that, Wnt signaling is inhibitory for oligodendrocyte differentiation. So natural next question will be, what's the TEM2 function in oligodendrocyte development and repair process? So I'm going to share with you, this is one gene that we study, how we study this gene in oligodendrocyte development. First, we actually make a knockout animal, specifically delete gene in oligodendrocyte lineage. So here we use SOX10-CRE line to delete TEM2 in oligodendrocyte lineage. And then looking at the mature oligodendrocyte expression labeled by MVP, and this is OPC markers. And this is a P0 very early stage of spinal cord. When you look at the conditional knockout of TEM2, you do see increased number of mature oligodendrocyte numbers, which is kind of makes sense. TEM2 is a positive regulator of the Wnt signaling. Now you remove that and then accelerate maturation. It doesn't really affect OPC numbers, which suggests that TEM2 function is very restricted in differentiation stage. So this is in vivo, but we also want to look at the cellular level. So we take these cells, print the culture dish. And as you can see here, the number of the oligodendrocyte is actually increased, but not only number increase, you do see morphologically they are much branched. So with that, culture and in vivo system, we thought maybe TEM2 knockout, conditional knockout shows much thicker myelination because it's much branched, more number, but it was not the case. When you look at CKO EM study at early development, you actually see very much impaired myelination. There's a lot of inner tongue structure. And then there's a reduced myelinated numbers. However, when you look at adult stage, this phenotype is almost, compensated. So this means TEM2 function in myelination is very critical for early stage, but it's okay for the homeostatic stage. However, given this early phenotype still maintained impaired motor function by Rotorado, this is adult animal behavior study. So this is like how we study with the little gene and specific oligodendrocyte lineage and looking at in vivo and in vitro study and beyond the behavior level. That's kind of how we study gene function. So now we know the gene TEM2 function. And next question is how that happens, trying to understand a molecular mechanism. So by the time we have done microarray to looking at specific gene expression in different development stage. Nowadays, no one does microarray, but this one has done early stage of my postdoc career. And then we overlay with RNA-seq and in specifically a lineage using the reporter animal. And then we also compare with the TEM2 IP mass. Basically, we want to identify TEM2 interacting molecule that are hopefully express oligodendrocyte lineage in the right development timing and associated with TEM2 by IP mass. So this is kind of how we narrow down the candidate. And if we find interesting angle, we will perform the functional study I showed you beginning using the mouse as well as the culture system. And this is kind of short list of those gene sets. As expected, we see a couple of Wnt-related genes. We also found cytoskeletal-related genes, which is both important for oligodendrocyte development. But very unexpectedly, we found E3 ligase, which is kind of a unusual genes, because E3 ligase function is pretty important for many other diseases, for example, neurodegenerative disease or even cancer. But myelin disorder has not been studied as much. So it caught our eyes, and we're trying to focus E3 ligase. By the way, those borders gene, throughout my almost 5-6 years in my lab, we identify as them to interacting partner has a unique function in oligodendrocyte. So for particular today, I'm going to share with you those two E3 ligase interaction with them too. So those two E3 ligase are very interesting. First, the VHL is a very important and critical tumor suppressor. And VHL function, it's a downstream HIF-1 alpha, is very well established in many cancers. But how VHL is regulated by upstream regulation is pretty much unknown. And another E3 ligase NEPO, which I will share with you more later, it's known to be interacted with SMARGT-based RET1 and another Wnt component TSHEV1. And also their lone function in cancer, but no one knows their function in the myelination. So we kind of target those two. And there's another reason. By the time that I know them to function, there's a nice paper published by UCSF group in Cell. They actually established VHL HIF-1 alpha pathway, which is known to be important for the cancer genes. And they actually regulate oligodendrocyte differentiation by Wnt signaling. And at the time, we identified them to interact with VHL and degrade, therefore promote gliomagenesis. So glioma and myelination is a completely different disease. However, we found a lot of similarity how they develop the pattern. So we kind of put together, sounds like it is possible TEM2 and VHL working together to regulate oligodendrocyte development and repair. So this sounds like a perfect project for the graduate student. So at the time when I start lab, I have my talented graduate student Hyun join the lab. So her project is basically identify them to function with a VHL if that's relevant in oligodendrocyte development. So we did a mouse study. We generate double knockout to try to compare. As I show you, them to conditional knockout shows more oligodendrocyte differentiation. VHL conditional knockout is very much impaired because we expected those two functions antagonistic. If you made a double knockout, this double knockout phenotype is actually reversed either single knockout, suggesting that TEM2 and VHL has antagonistic relationship. It doesn't really much affect oligodendrocyte progenitor cell, still put TEM2 and VHL axis function in very specific for the differentiation stage. We also confirmed complementary gain of function studies using primary oligodendrocyte cell culture. Basically, we overexpress TEM2 that inhibit differentiation labeled by this green cell MAG. However, if you overexpress TEM2 and VHL together, VHL actually antagonize TEM2 inhibitory function. So this gain and loss of function study really suggests that TEM2 and VHL is antagonized in oligodendrocyte development. And this is good, but TEM2 is another E3 ligase. How does TEM2 degrade VHL? Must be something recruited by E3 ligase. So we look at our gene set, and then we did biochemical studies, all of them, and then we found NEPO is a potential one. But when we did a biochemical study, we used a HEC293 T cell. It has very nicely degraded by NEPO, but it's very interestingly, if you compare HEC293 T cell versus oligodendrocyte specific cells, their biochemical phenotype is very different. And actually, we found that NEPO stabilized VHL in oligodendrocyte lineage cell by K63 chain modification. So if this is true, we need to confirm our in vivo study. So we make a NEPO conditional oligodendrocyte specific conditional animal. It really showing that loss of a network in oligodendrocyte, impaired oligodendrocyte differentiation by mature marker expression, PLP and MVP. And by EM studies, if you remove the network in oligodendrocyte, it's really, really impaired myelination. So suggesting that NAP4 does really stabilize VHL because their phenotype is parallel and antagonize TEM2. So this is like one example how we study TEM2, one gene and their downstream. So over 6 years of my lab, we identified TEM2 and antagonistic function of VHNF4 in oligodendrocyte lineage, specifically affecting in differentiation stage. And I didn't have time to bring all the data, but we also show that TEM2 is actually working on early stage interact with the backbone to controlling AKN assembly because it makes sense, it's booming protein. In the later stage, in the maturation stage, it tends to work with zessolin, which is a critical component for myelination and controlling the proper myelination. So this is like how we study gene function downstream, all the development stage. But we are also interested how this development observation is relevant to human disease, how these pathways involved in human disease angle. So as a starting point, we first look at gene expression in human disease. As I mentioned, in humans, different age group has a different disease. In newborn baby, the hypoxic condition called hypoxic ischemic encephalopathy, these babies shows very poor myelin structure integrity, and it's known that oligodendrocyte is most sensitive under apoptosis at first. So we look at TEM2 expression in oligodendrocyte lineages in this newborn HI sample, found that TEM2 is highly expressed, suggesting that TEM2 is antagonistic function in differentiation, therefore is highly expressed in the lesion of HI sample. And not only newborn baby in the sample, we also look at human MS patient sample. And it is a little bit hard. We have a quantification for the publication purposes. It is significantly increased TEM2 expression in oligodendrocyte lineage. So we first always look at the gene expression in disease angle. If they are expressed, then we're trying to manipulate in the mouse model. So for specific disease, we use a specific mouse model. For the MS, we use 3 different mouse models, lysolecithin, EA, or coproison. So today I'm gonna share with you some of the data from the lysolecithin. And for the HIE, we use hypoxic injury chambers. Basically, we put the babies like pop from P3 to P11 with a mom under in vivo hypoxic chamber to mimic the babies under hypoxic condition. So we kind of use a different mouse model that depends on disease condition. So for the sake of time for today, I'm going to share with you one of the model, lysolecithin model, how we studied them to function in adult white matter injury. So lysolecithin is a kind of a chemical detergent. It actually melts the myelin and they ultimately kill the oligodendrocyte. When you inject the lysolecithin in vivo, either ventral white matter of the spinal cord or corpus callosum of the brain, you do see over time it's nice demyelination and remyelination. It shows very dramatic dynamics of repair process. And this time course in the very early stage, oligodendrocyte precursor cells recruit into the lesion area and start to differentiate it, and then finally at around 2 weeks in spinal cord, they fully repair. So this is a well-established mouse model. So take this model, we can study loss of function study of them too using knockout animal. And here I use a whole body knockout. Basically, if you inject lysolecithin in the animal, they're looking at 7-day post-lesion. 7-day post-lesion in control is barely differentiated, barely repaired, as you see here. However, if you delete TEM2, it's very much increased repair process, suggesting that TEM2 is really antagonized. If you remove that, it accelerates a repair process. Vice versa, you can do also overexpression study using viral approach. You can inject them to virus to see inhibitory function during the repair process. Of course, you can do lineage-specific conditional animal to confirm this phenotype is very specific to oligodendrocytic TEM2 expression. So that's how we study. And I don't want to bring all the data because it would be too much. So I will probably skip. Basically, simply we can do those TEM2 and VHL axis function using genetic mouse model, same approach. And this is kind of a quantification. TEM2 conditional knockout has a high number of PFP differentially cells. If you make a double knockout after injury, this phenotype is reversed. So this is a kind of one disease model we study. We have performed a different disease angle to find TEM2 VHL axis with NEP4 has really dynamic antagonistic function even in the repair process. So this is like a first part that I show you that I bring the one gene called TEM2, how we study TEM2 function in oligodendrocyte development. We can generate specific mouse model, and then we can also do primary culture cell to do more biochemical studies, and then perform the in vivo behavior study, how this structural function is affecting the mouse behavior, and complicated those downstream pathway we can always study with generating double knockout animal or manipulation in vitro. And what we found for 5-6 years of work, we actually found a lot of similarity for the myelination as well as tumorigenesis, because a lot of genes are shared, and this is one angle we're trying to define shared or unique pathway in myelination as well as tumorigenesis. So this is like one part of my lab is heavily working on. And I told you the Wnt signaling is such important, but I didn't really say about the Wnt signaling in TEM2 and oligodendrocyte. It's very complicated, but I have to confirm it. This is really due to the Wnt signaling. So that's what currently one of the postdoc is working on it. And just brief taste of how Wnt signaling is regulated in oligodendrocyte development. It is very dynamically regulated. However, if you look at the beta-catenin signal in developing CNS, more developed mature stage has high expression of beta-catenin in the nucleus, meaning Wnt signaling is more active in the mature stage of oligodendrocyte. When we look at TEM2 expression, TEM2 is highly expressed at the later stage, mature stage of oligodendrocyte. And this is quite odd because TEM2 is inhibitory factor, it's partial regulator. Beta-catenin expression and TEM2 expression is correlated during the development. But why they are high in the mature stage? So that's a starting point. And I didn't bring all the data, but hopefully soon we can have published an article for my postdoc's work. We found that it is a true mature oligodendrocyte TEM2 is more expressed. Beta-catenin is more active. However, this dynamic expression is tightly controlled by phosphorylation of TEM2, which lead to degradation of TEM2 with kinase and E3 ligase. It's a very molecular detail pathway, but this can be explained why Wnt signaling is actually required for fully mature myelination stage, why TEM2, even though its inhibitory role in oligodendrocyte, its expression wise high in mature oligodendrocyte. So I didn't bring for today, but hopefully in a few months, we can have published a record for this study. So this is basically two talented graduate students and one postdoc's work in oligodendrocyte development and disease angle. And those two graduate students now all graduated, and postdoc, hopefully he wrapped up his last project, and then he is actually got already faculty job in Taiwan. It's going to move out. There's a new step. So Then who's going to working in my lab? We have half of the lab is working on another type of the glia cell called astrocyte. I'm going to just show you what we study, not really science. That way we can discuss if someone is interested in astrocyte biology. So this work is done by my Korean postdoc Chuyan and my second postdoc Chi. So astrocyte is very interesting. They have a unique interaction with the neuron to form the synapse. They also have a unique interaction with the BBB. So we are very interested how this astrocyte make this unique interaction with two different cell types and how that function is affected in development as well as a disease condition. So first, I will share with you them to function in astrocyte with neuron interaction. So first, we want to make sure that is expressed astrocyte. So with the reporter, we found that is a highly expressed astrocyte throughout the development. And then we make them to specific knockout in astrocyte. What we found, if you remove them too, we do have really morphologically big astrocyte. And this bigger astrocyte is actually presented throughout the brain region, not like a specific one brain region. But this morphologically bigger astrocyte has very different function in different brain regions. This is unexpected. So for example, in cortex, this loss of them to an astrocyte could really reduce astrocyte calcium activity. However, in olfactory bulb astrocyte, it's increased calcium activity. So this is a very unexpected. We confirmed many times, but it's continues showing this different phenotype. Then we wonder what about neuronal activity? And then it really shows different neuronal activity. For example, in cortex, there's increased neuronal excitatory synaptic current. However, in the olfactory bulb, inhibitory cells are more active. So we are very kind of stuck there because we only manipulate them to an astrocyte. And they morphologically bigger in both brain regions, but their outcome, functional outcome is very different. So we did screening, we did a Mass-spec screening in two different brain region area. Then we found this much gene set differences. And there's 13 gene is actually unique shared. And if you look at this heat map of these 13 gene, their expression is very diverse compared to the cortex and olfactory bulb. So we actually picked one of gene and then see the functional study. But we found that, for example, SAC4A4, which is channel, pH channel specifically expressed astrocyte. This gene function in them too is very specific to cortex. However, olfactory bulb or cerebellum tend to function with the SAC4A4 is not really established. So that's kind of how we do the study. And another part, we are also interested in astrocyte and BBB angle. And because we had lots of genes that screened from the beginning, we found one gene called SAC4A4, which is a highly expressed astrocyte as well as this vascular astrocyte. And this gene function in astrocyte has been studied heavily in vitro system or ex vivo system, but not really studied in vivo setting of the astrocyte function. So I don't have time to go over all the detail, but what we actually found these days, we found if you remove SAC4A4 specifically in astrocyte, it really increases chemocytokine expression that cause really impaired BBB in development. And we also apply this function in the ischemic stroke mouse model, and it shows a really dramatic BBB impairment at the ischemic stroke. So this is kind of a new story that lab is working on, and astrocyte neural interaction we already have published, but astrocyte-BBB interaction, my another postdoc is working on. Hopefully that also soon to publish. So this is kind of what we study for the astrocyte. So I hope I can make some take-home message today for the oligodendrocyte development and repair. I share with you one gene called DEM2 study, how we study this gene function using genetic mouse model in vitro culture biochemical studies. We mainly focus on downstream study, but currently lab is working on them to upstream regulator using post-translational modification method, modification pathway. And we also study astrocyte, and again share with you time to function in astrocyte. Also, I share with a little bit of the assays for function in astrocyte Astrocyte-BBB interaction. So it doesn't really have a really depth of the science, but I hope this talk will give you an idea of what my lab is studying. And this is my lab currently. 2 graduate students graduated, and then I have a new one started. And then my 2 postdocs got a faculty job, and then they are moving out. So I would say right now it's kind of second generation of the laboratory. And we heavily study more like a molecular-centric cellular molecular biology. However, in the second generation, we're trying to study a little more conceptual advanced topic, which I don't have time to show today, but I will be happy to discuss in the discussion section. And this is my funding and collaborators. And as Dr. Kim mentioned that I am in this Neurological Research Institute, fully built by donor funding. So Baylor is right adjacent, and then Texas Children's is this side. So I'm in the middle and a dual appointment for studying my laboratory. So this is like how my lab does. I'm happy to take any questions.